welcome to my channel. This is the second video of a series called Meet the Pros. In this series, I will be interviewing people in the field of biology and genetics to help high school students interested in biology. In today's episode, I will be interviewing Dr. Ramanarayanan Krishnamurthy. He is a chemistry professor at the Scripps Research Institute in San Diego, California, and I'm delighted to have him with me here today. Thank you for being here,
Thank you.
Dr. Krishnamurthy.
Yeah, thanks for this nice opportunity.
Yeah. So I want to start off with a basic question. What got you interested in the sciences, especially chemistry?
I would say I think it was the chemistry teacher, probably the high school teacher, Mr. Prabhakar at Vidyamandir High School in Chennai, who was very
Okay.
good and who got me to appreciate molecules. And for me, I think from a young age, I think I was very fascinated by how molecules coming together can give rise to more complex things and give rise to a property which is very unique, what we call life.
Mm-hmm.
And it's not just a mere collection of chemicals, but the way they are put together, as Carl Sagan would say.
Mm-hmm.
And my interest in chemistry and science started with these kind of questions of why things are the way they are. Why would they not be different?
Mm-hmm.
What controls them to be this way and not the other way?
Yeah.
And those kind of questions began to really, and for me, the answers seem to lie in understanding chemistry, which is fundamental to probably biology as we know it today.
Okay. So do you have any advice for high school students interested in biology or chemistry, specifically like things that they could be doing outside of just the school courses?
So one of the things that I see is that people who are quite successful, especially young people who want to come up, they take a lot of initiatives of their own and they follow their passion.
Mm-hmm.
And what I would suggest is that if you have a passion that is coming from the school courses that you take, you can take that up further by either reading books or contacting people with whom you think you will profit by making those connections. Say, for example, you may have a question as to why things are the way they are, or you might have heard something in your class about a chemical or a biological principle and you want to know more about it. Now you have the internet, which brings information at your fingertips, and then you can make connections. So I would suggest that they broaden their mind by either reading or contacting people who are the prominent leaders or their local colleges, professors, to see what sort of opportunities would be available for them to expand their horizons.
So it's all about being like open-minded and looking at what's available for you.
Definitely, definitely, because in science, one of the things that is certain is that there is no one final answer to these things. You keep changing things as things move and new information comes up. Of course, the fundamental laws do not change,
Yeah.
but the consequences of that might change depending on the conditions and the environment they are manifested in.
Okay. So now I want to switch over to a couple science-related questions. Um, could you talk about your work on the origins of life?
I was hooked onto Origins of Life by a fascinating lecture by my mentor, Professor Albert Eschenmoser of ETH, Switzerland, when he gave a lecture at the Ohio State University, where I was doing my PhD in chemistry in 1991.
Okay.
And his question was, why did nature choose the type of molecules it did for, RNA? So when you go talk to people and ask them the question, what is the first molecule that comes to your mind when you say sugar? People will say sugar, glucose, fructose, sucrose,
Mhm.
but rarely people will say ribose. Ribose is also a
Yeah.
sugar, but it's
Yeah.
one of the most important sugars in RNA.
Yeah.
And the question was, if nature had the choice of sugars, why did it choose a ribose
Mm-hmm.
among all the other sugars? And then why did it choose a five-membered formation, a pentose, a
Yeah.
ribofuranose
Yeah.
to make that? So the question was, so that's what got me interested. That means you can And as an organic chemist, I can begin to make the other alternatives, come back and compare, and then try to understand what is so special about ribose. And one thing that is very important for people to recognize is you do not recognize or understand why one thing is so special by studying that only. You need to make a variant, you need to make something different, see what changes, and come back and compare. And that's what organic chemistry
Yeah.
allowed us to do.
Okay. So you mentioned RNA and how it started off with ribose, I believe. And
Yep.
this was something that we both learned at school and that I saw in the podcast that you sent me. And
Yep.
I was wondering, what do you think about all of these theories that are there in science about the first molecule? So the RNA world theory, the protein world theory, lipid world theory, things like that. What is your take on that?
Okay. These are all very good hypotheses. In science, you always need a hypothesis to test it out, and you need to try and falsify it as much as possible. And if you fail to falsify it, you begin to understand the rigor of that hypothesis a little more, and you begin to then can fashion experiments to prove that hypothesis. Now, these RNA world first theory and protein world first theory and all these theories that you see around, origins of life are all based on what we understand life to be today. So we know life to be based on RNA, DNA, proteins, lipids. And so the idea is to look for those kind of systems back about 4 billion or 4.2 billion years ago on early Earth. And for me, that is a kind of a problem, because so many billions of years ago, such sophisticated molecules were not available. So when you're looking from bottom up and you do not know what your final molecules or final polymers of life are going to be, how do you already justify the components of those polymers in a prebiotic sense? So what I try to do is try to remove a purposeful evolution, if you will, and just let the chemistry and the science teach us where systems will move. And you may come up with a very different answer if the environment was totally different. So ask yourself this question: if you land on an exoplanet, and
Hmm.
James Webb is finding so many things now,
Yeah.
and that environment of that planet is very different from what we have. Water is not the solvent, but a different solvent,
Hmm.
silicon based materials.
Yeah. Yeah.
Would we get to the same type of molecules that we think on Earth today? So that is how I would like to approach this problem: completely agnostic, not knowing what the solutions are, not knowing that RNA might be a molecule, and see where we would end up if we start from ground zero.
Okay. So basically, it's like what conditions were there on early Earth, and then what happened to get to where we are right now, the foundations
That's
of.
right. And that is the huge challenge, because the
Yeah.
early Earth environment, there are a lot of hypotheses again about it. We
Hmm.
have some fairly good idea about certain things, and we have completely no idea about certain other things. For example, we know what sort of bombardments might have taken place,
Hmm.
whether there was a land mass enough available on early Earth. Earth was covered by completely water at a certain point. So were there land masses to allow the chemistry to take place, certain types of chemistries, and those, we are able to put certain restrictions of this is what would be available, this would not be available. As to the molecules themselves, what would be the building blocks? We know there was a lot of carbon dioxide, a lot of nitrogen, but how do you convert them down to the types of organic building blocks that you would begin to? And there are again a lot of hypotheses and a lot of ifs and buts, and certain conditions have to be met. And so people are very good at coming up with hypotheses and working with it, but trying to take it back to what would have been exactly on earlier remains an open question.
Okay. So then, if there are so many ifs and buts, as you said, about the early Earth conditions, then how do you go about designing your experiments in terms of whether or not your simulation of that experiment could have been possible on early Earth?
Okay, that's an excellent question. So let us say, even if I did come up with an experiment where I start mixing molecules and put it in a beaker, and lo and behold, about a month later, life crawls out of it. Your
Hmm.
question would be, yes, you did demonstrate it, but what proof do you have it happened the same way 4.2 billion years
Yes.
ago?
Yes.
And for that, there is no definite answer. So what we try to do in this field is we try to remove the historical perspective to a certain extent so that we can form a hypothesis and do chemistry and do the experiments. And then we say, okay, under these conditions, these are the type of reactions would happen, and these are the type of molecules would form, and these are the type of chemistries which would lead to the biochemistries that we see today.
Mhm.
That's the best we can do with the knowledge that we have now until we find chemical fossils, of a ribose molecule that is 4 billion years ago, or DNA that is about 4.1 billion years ago. That is going to be impossible to answer,
Mhm.
like the fossils that we have. Right now we know about dinosaurs, we know about these kind of things because we rely on fossils, and we know they came from much simpler unicellular organisms because of the fossil record. Unfortunately, we don't have a chemical fossil record equivalent by which we can walk back. The earth has destroyed it because of its diagenesis, and life itself, which is now today, is so prevalent that
Yeah.
even if you find something old, you have to prove that it is not contaminated by life or from life that is living, available today on early earth.
Okay. So a lot of the traces of what early Earth was like are basically wiped out because of what life is like today.
The solid ones that you would need
Yeah,
to
yeah,
say,
yeah.
okay, this chemical. But you can have, for example, there are some zircon rocks that you can go and you can rely on. You can quantify how much, what the temperature would have been, how much water would have been available, what sort of process went on. All those geochemical constraints you can put on it. But when you come to the organics, that's a little more on shaky grounds.
Um, so you mentioned how we have a fossil record, but we have no chemical record, right? What would it
Yep.
take for us to have a chemical record that goes back as far as maybe at least like 3 billion years?
Okay, so there are some old fossils people have found in Australia. They have pushed it to about 3.2, 3.4 billion years. But beyond that, but even those are already highly evolved life forms, if you
Yeah.
will. So
Hmm.
if you want to go even beyond that to what we call a prebiotic chemistry, even before the life forms were formed, that is what we rely on is just carbon. So you might have heard of carbon dating based on the
Yeah, 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 yeah.
carbon isotope C13. So what people do is they rely on this enrichment of C13 isotope versus the C12 because life uses the lighter version C12, and they try to find out how much of the C13 to C12 ratio is available. And what they find is some, because organic matter decomposes and finally goes back to carbon. They rely on this carbon dating, if you will, to find out the ratio and say, oh, this must have come from an organic source, this must have come from an inorganic source. That's the best we can do at the moment and not go any beyond because that's nothing but carbon molecules put together like graphite, if you will. And there is no other piece of information that you can glean through by studying that, at least with the techniques that we know now and we have today, the state of the art, to say it must have come from a DNA or it must have come from a protein.
Okay. So then, based on everything that we do know right now about what early Earth could have been and what it is like right now, could you speculate on any similar chemical reactions that could occur on Mars? Like things that could maybe contribute to the possibility of life surviving there?
That's a fascinating question because it is believed, and there are quite good number of reasons to say so, is that early Mars had almost the same or similar composition and type of atmosphere and chemical availability of early Earth. And that's why Mars people are very fascinated about Mars. They would like to go and see, and then Mars has not suffered the same type of volcanic activity, if you will, or some sort of diagenesis the same way Earth has. And so they believe that some of the records might still be intact on early Mars. They can, if they can dig deep and things like that. That's why you see so many NASA missions to
Yeah,
Mars, or
yeah, yeah.
they are really trying to find out if there are organics, if there, if can you study those organics, can you get more information from those organics that you could not on Earth, and things like that. So there is a group highly dedicated group studying the possibility that early Mars could have had life
Hmm.
very similar to what would have been on Earth. And so, but again, the jury is still out on that, and we have had many false, I would say, starts with claims of life on Mars, or claims of lifelike things on Mars. So it's still on an ongoing process, and people are very careful now in terms of trying to go through in a very rigorous manner before they try to make any claim on that.
Okay. I was also looking at your lab's homepage, and it referred to creating artificial informational polymers. Could you talk about that and like what they are?
Definitely, definitely. So the information polymer that biology relies on is DNA and RNA,
Mm.
and a certain amount of protein encoding that takes place. So what we are trying to find out is, if you look at DNA and RNA, they are made out of the same type of backbone. So the
Yeah, yeah.
deoxyribonucleosides and the ribonucleosides.
Hmm.
And the question for us is this the only type of backbone with the four nucleobases AUGC in RNA, or the ATGC in DNA? Are these the only combinations available? So in our lab, what we have tried to do is try to do a Lego building block combination, so that we take a building block of DNA, building block of RNA, a building block of an artificial nucleic acid. Say it might be a thiofuranosyl nucleic acid, what's called a TNA, or a ribonucleic acid, which is ribulo and ribose are isomers of each other. And so we have made all these different building blocks, and we are now trying to mix and match them together. So it's like a yellow building block. So if you think of DNA as completely yellow building block
Yeah, yeah.
backbone and RNA as a red building block, we are trying to mix them, match them, and see if we can come up with a different paradigm. And
Mhm.
we have answers. Yes, indeed, you can. And so that is going to be one of our major projects going forward, is that what sort of different backbones and different bases we can use and come up with completely different um, alphabets, if you will, of an artificial nucleic acid, and whether that would be useful in a certain way, either to be put back in biology, or for material chemistry, or for information encoding purposes. So everything is possible with these kind of artificial backbones, artificial nucleobases, and things like that.
So, what are these new paradigms that you're creating based off of, I guess, making these Lego blocks?
Yeah.
What does that teach you about the early Earth conditions? Because if they are artificial, then they're made with the foundations of life as they are right now,
Correct.
right?
So what we are trying to find out is life's building blocks and life's paradigm the only possible solution on Earth? So if we come up with an alternative building block, alternative paradigm for making a life form, if you will, the question is, what sort of conditions would you need to make that life form be the predominant one and not the one that we know today? So it's a question of, it was famously asked, I think, by Jay Gould from Harvard, who said, if you play the tape, if you rewind and play the tape again of life, would you come back to the same answer? And my take on that is that if you change the conditions, for example, right now we need water, a temperature scale between 0 to 100 degrees, and a pH, a physiological pH, which is close to 6.7 to 7.
Mm.
If you change those conditions, then your answer is going to be different.
Yeah.
If you are going to be always acidic, then the type of biomolecules that we have today, like RNA and proteins, will denature and will not function. Therefore, you have to come up with a different molecular solution that can function under a pH of around, say, 4, or a
Yeah.
pH of 11. So
Yeah.
that is what we are trying to find out. What are the conditions under which you will get alternative building blocks, which may lead to a totally different answer than what we see today on Earth, on Earth.
Very interesting. Yeah.
Yeah.
So finally, I want to end with, I guess, a fun sort of question.
Yep.
If you could bring back any extinct species, which one would you bring back and why? And you cannot say dinosaurs.
Okay. I have a more of a philosophical twist on it.
Hmm.
I would rather not bring back anything that went extinct. simply because it went extinct for a good reason. Either it could not
Mhm.
survive the
Yeah.
change in conditions. For example, 2.5 billion years ago on Earth, there was not enough oxygen.
Hmm.
Suddenly there arose oxygen because
Yeah,
of,
yeah, yeah.
so what happens is, because of the rise in oxygen, a lot of things went extinct that could not survive that.
Mhm.
So the question is, why would you want to bring that back into this oxygen rich environment
Yeah.
and make it, I won't say suffer, but make life difficult
Yeah.
for it, if you will.
Yeah.
So, so the reason some things went extinct are because they could not survive, and so it would be probably best to leave them as what they are, and then go on forward. That means you probably, for me, it is, you look at the past with an idea of how you can take the present to a better future, rather than trying to revive that one that went off, extinct. What can we learn from that to make us not go extinct, if you will?
Okay. So then is there anything that you would do to, I guess, make this future better in a sense, or anything that you would wish for?
So one of the things that we are forced to reckon with is this climate change, if you will, right? There is
Yeah, yeah.
this lot of. So the question is, can we begin to come up with chemistries that would begin to use this waste material that we are producing back into something that could be
Mm.
recycled? So
Yeah.
carbon dioxide is one of them, and that has come to a forefront interest for us, because carbon dioxide is the source molecule for sugars. That's
Hmm.
how nature makes sugars. The question
Yeah.
for us is, can we come up with procedures that would try to make the sugar molecules, convert carbon dioxide to type of sugar molecules that would be useful for consumption, and that way we can begin to sequester the carbon dioxide. So that is how I look forward to that kind of an opportunity within
Yeah,
my
yeah.
research group to
Hmm.
tackle that kind of questions.
Okay. All right. Well, I think that's all the time we have for. Thank
Yeah.
you, Dr. Krishnamurthy. I really appreciate you joining me today.
Thank you, Rishabh. I really enjoyed it,
Me
very
too.
much. Thank you.
Thank you.